Good morning, everyone. Thank you for attending our webinar. I will be discussing and demonstrating our implementation of matched molecular pairs that will be available in the next version of Medkin Studio, which is set to be released in March next month. This next slide is simply an outline of the talk, and we'll start with an introduction to matched molecular pairs. In the second section, I'll discuss using matched molecular pairs for exploring SAR and designing new molecules. In the third section, I'll discuss improving solubility using matched molecular pairs. So in this case, I'm going to use solubility as the property. And then finally, in the last section, I'll describe using matched molecular pairs for predicting off-target interactions. So what is a matched molecular pair? It's simply a pair of molecules with a small specific structural transformation. So if we look at these two molecules down at the bottom, the first thing we want to do is identify the maximum common substructure. And in these two structures, it'll be the pyrimidine ring, the sulfonamide, and the aniline group. And what's left is the small specific structural change. So in this case, chlorine is being replaced by a methoxy group on an aromatic system. So let's say we were interested in trying to create matched molecular pairs for solubility. Both these compounds would have experimental solubilities, and here I've expressed those as the log of the solubility. So the larger the number, the more soluble the compound. The change in solubility, going from the compound on the left to the compound on the right, is 1.615 log units. So you're increasing the solubility by over an order of magnitude and a half, approximately. Okay. So if we took a large number of these pairs using matched molecular pair analysis to automatically identify a large number of, or a number of matched molecular pairs, and then analyze the change in property between the values. Obviously, if you replace a chlorine with a methoxy on an arbitrary molecule, you're not going to get this exact change in solubility, but you'll get a distribution of changes in solubilities, and that's part of the analysis phase. If you take the average of these compounds or pairs that have changes in solubilities, then that would be the expected size of when the transformation is applied to an arbitrary molecule. Okay. And if you look at outliers from this distribution, you might see compounds that maybe buck the trend. In other words, maybe replacing a methoxy with the chlorine might decrease the solubility in this example. Okay. There are a number of companies that have published articles on matched molecular pairs. A lot of them are large pharmaceutical companies such as AstraZeneca, Merck, Pfizer, Abbott. And one reason they're able to do this is they have large data sets of various properties. There are also universities such as Sheffield, Bonn, and University of California at San Francisco that have recently published papers on matched molecular pair analysis. So what types of areas is this can be applied to? Well, I just showed a brief example of aqueous solubility. I'll show more of that in the second section. One of the examples I'll show in the next few minutes is of ligand potency. People have also looked at bioisosterism, plasma protein binding, oral exposure, intrinsic clearance, hERG and P4 binding and P450 metabolism, as well as drug repositioning and side effects, and then finally patent protection. Okay. So how do we calculate the similarity between a pair of molecules using a maximum common substructure? So first we identify the atoms that are similar in both of these groups, and that would be the 34 matches between these two pairs, and simply divide it by the total. And the total is the number of matches plus the number of mismatches. So for this pair here, we have a similarity index of 0.65. So it's a fairly intuitive and simple calculation just based on matched and mismatched atoms and bonds. Let's not forget about the bonds. We align the cores to increase the interpretability, and I'll show a brief example of that on the next slide. You can also modify the rules. So you could say maybe an aromatic carbon is equivalent to an aromatic nitrogen. We've modified the maximum common substructure to allow disjoint cores. So, for example, maybe this gets into the sites of different areas of molecules that are changed between the two pairs. And by allowing disjointed cores, we can perform similarity searches on a little bit dissimilar compounds, and I'll show examples of that in the very last section. It's a little bit slower compared to just calculating fingerprints and Tanimoto coefficients, but we've implemented this in multi-core as a multi-core process, so that will speed up the computation. Okay. We also align the scaffolds based on the maximum common substructure. So these two, there is an option to either align by redrawing the structure or align by just rotating the structure. And so this alignment here that I show on this slide is simply the alignment by rotating. If we were allowed to or let the program align based on redrawing the two molecules, we see a much more intuitive overlay of the two compounds. So in this case, the imidazole would kind of match up with the phenyl ring here. The methylene is aligned on both sides, along with this lactam area of the two rings here. And then finally, we have this phenyl ring over on the right-hand side molecule, matching the phenyl ring of the tricyclic group over here on the left-hand side molecule. So this kind of allows you to kind of pick out the common substructure a little bit better. Okay, so let's go to MedChem Studio and just show how this is implemented. And what we're going to look at here is a set of 12 CDK4 inhibitors from a publication in 2005 from Journal of Medicinal Chemistry from Too Good. So all of these compounds are CDK4 inhibitors. I have a column that is the IC50 in nanomolar quantities for CDK4, and then I also have a column that is the PIC50 value of these compounds. So the higher value here would indicate the more active compounds. Okay, so what I can do is combine all these into a single class, and then if we look at, I'm just going to circle what's the large common core in these molecules, and then go over to the R group pairs analysis, and you'll see that we have an R2 substituent and an R1 substituent, and the R1 is simply a bromine and an acetyl group, and then we have 6 different R2s. What we're going to do is create matched molecular pairs looking at the PIC50 as the property value for these. Okay. So we simply click on the molecules pair tab, and we have 3 options here to specify. So the maximum number of atoms and bonds that are on a match, we'll leave that at 20. The minimum Tanimoto at 0.65. We'll also leave that as the default. And then we'll change the maximum number of sites in a pair to only a single site. Okay, so this will only only show us differences in one site on the molecule. So very quickly, we generated 22 pairs of molecules according to that criteria. So if we look at this first pair of molecules, we see that essentially we've taken a morpholine and replaced it with the pyridine. And the activity of the first compound is 5.7 for the PIC50. The second compound is 6.796, and the change is about 1. So going from the morpholine to the pyridine, you've increased the activity by an order of magnitude. If we look at our next pair, we've replaced a bromine with an acetyl group. Here we've gained an activity change of over 2 orders of magnitude for this change. So the first step in the analysis is identifying these pairs that only differ by one site. The next phase is to cluster these pairs together. And so we have a couple of different options, but I'm going to select cluster together pairs with the same structural transformation. So again, I click OK, and we come up with another option that I'll discuss in the next section. So I just use the default, click OK, and it's created 9 clusters of matched molecular pairs. So in this first cluster, you can see highlighted in the molecule on the left is the bromine atom, and on the right is the acetyl group. So that's the substitution. There are 6 pairs of molecules of which these have made, and a total of 12 molecules. This chart represents the distribution of the PIC50 for all 12 molecules. The second column is the change in activity in going from replacing the bromine with an acetyl group in this case. And then finally, another column I just want to show is the average change. So of these 6 pairs, the average activity increase is 1.24 log units. Okay, we can look just brief at some of these other pairs. So in this case, N-methylpiperazine is replaced with 4-hydroxypiperidine, and there's 2 of these pairs, and each one of these pairs is simply one is the bromine shown here, the other will be the acetyl group here. Okay. We can also just simply click on the pairs tab, and this will show the 6 pairs associated with that specific transformation. Before I do that, let me just briefly show this other column, which is the SMERCH column. Okay, so we've encoded this transformation in a SMERCH string. So this basically says that you take a bromine and replace it with an acetyl group. And then we can save these transformations and apply them to other molecules. And I'll talk about that when I get to the slides. Okay, so now I've gone back to the clusters. So in all of these, we're replacing a bromine with an acetyl group, and they're ranked in order of decreasing change. We can invert that to so that the change is in increasing order. And you'll see here that when you have the 3,5 dimethylpiperazine, that the change is not as big. So even though on average replacing this is an order of magnitude difference, you also need to kind of consider the left-hand side of the molecule in this case. Okay, and then finally I want to show our networks viewer. Okay, so by default it just displays 1 pair initially because the screen can get a little bit crowded, but if we click on or hover over the bottom molecule, we'll get information about the activity of that molecule shown in the upper left-hand corner. If we click on the red square, we get the difference between the pairs, so the difference in activity is going up in the right-hand corner. Okay, and then finally we can show all of these transformations or display all of these transformations in the network map. So along the y-axis, this is the PIC50 distance. So the higher up on this panel, the more active the compound. And we can move these molecules around on the horizon. Okay. So now let's go back to the PowerPoint slides. Okay, so now I want to talk about activity cliffs in HIV integrase inhibitors. So matched molecular pairs can also allow you to identify activity cliffs in molecules. So in this case, we've taken a set of HIV integrase inhibitors and generated pairs, and in this case, 2,312 pairs were identified. We can also graph the change in IC50 value versus the similarity index. So you would expect that compounds up in this circle up here are compounds that have a high degree of similarity, but they also have a large change in activity. So these are activity cliffs, compounds or pairs where you've made a small change in structure, and that's manifested itself in a large change in activity. And that's kind of an unexpected event, but something you want to be aware of in order to identify maybe groups that are kind of a warhead are very important for activity and really boost the activity up. Okay. One of the pairs identified in this process was this pair shown above, and in this case, hydroxyl group is actually being removed, and that is accommodated with a change in activity of over an order of magnitude in activity. So by removing the hydroxyl group, we've increased the activity. This change can be represented as a set of transformation rules, and in this case we've kind of expanded the context of where this hydroxyl group is, and it's in the ortho position relative to this group. And the two SMRP strings just represent different ways to identify this group. In the top case, we're looking at this ethyl group. In the bottom case, we're including this carboxylic acid functional group. Then we can take these rules and apply them to existing compounds to generate novel compounds that are at least predicted to have increased activity. So if we took this compound on the left, which is already in the data set and has a known activity of 5.8, and remove this hydroxyl group, what kind of activity would result? And if it's a similar SAR to this one on top, then you'd expect an increase in activity. Okay. And this next slide simply shows the pairs of molecules on the network viewer. So this is our first pair, the known pair, where we're removing the hydroxyl group. It increases the activity as observed by the, it shows up higher on the vertical axis. Here was the other molecule that's measured, and if we remove this hydroxyl group, what kind of activity are we going to get from that? Okay. So also in MedChem Studio, we have a way of applying transformation rules. So this was already built into MedChem Studio, and it simply will take a set of a single compound, generate a number of analogs based on rules. And these rules can either be bioisosteric replacements, or rules from literature or in-house experience. And we could also add rules derived from a matched molecular pair analysis. So you would want to apply rules, though, that for your particular target, if it was some other property, say solubility, you could apply that more globally. Okay. So now in the second section, I want to switch gears and discuss using solubility as the property to evaluate. And so again, I'm going to start with a demonstration. Okay, so here we have 3,000, roughly 3,000 molecules with experimental solubilities in milligrams per milliliter. And then I've also added log solubility, etc. So I'll go back to the classes tab. So now we have 3,000 pairs in here. And I've already calculated the molecular pairs for all of these molecules, and then save the CTK file. So it'll automatically save the matched molecular pairs that I've generated, and I've generated 10,586 pairs that were saved on disk. So let's say I want to kind of simplify this and only look for specific transformations. So I can go down to the reaction query dialog box, and I'm going to just type in, you could put this into a sketcher, but I'm going to just type in a SMRP string, and basically this says if you find an aromatic CH group that's going to an aromatic carbon attached to a heavy atom, then highlight that, that will match the query. And I'm also going to only allow exact matches. So this asterisk here represents a wildcard, but it's only going to represent one heavy atom because I'm only allowing exact matches. So I simply click OK. That results in 982 pairs selected. I'm going to hide the unselected pairs and then create the pair clusters on these 982 compounds. And again, I'm going to cluster together pairs with the same structural transformation. Okay, so let me talk a little bit about this option here. So by default, we only look at a very local environment around the change. So in this example here, we're only looking at this carbon atom and this carbon and hydroxyl group. We can require that the program go out and search a larger area of this a scaffold so that you're being much more specific and specifying the structural context around the change. So we'll talk about that in a few minutes, but right now I'll just go back to the default. And so it's comparing all of these 982 molecules and clustering together the ones with the same structural transformation. And this should finish in a second here. And then pops up into the pair clusters tab. Okay, so here we have 8 clusters of compounds. This first one is simply a CH going to a carbon chlorine bond, aromatic bond, and we have 461 pairs, a total of 323 molecules. This is the distribution of the log of the solubility in molar quantity, and then this is the change. And as you notice, just adding a chlorine to an aromatic ring decreases the solubility, and the average decrease is 0.628 log units. So we'll get back to that one. Here's another change. Now here it's going from a methyl group, so you're removing a methyl group in this case, and removing a methyl group here increases the solubility of the compound. So in this case, it's matched the equation going both ways, so which is actually quite a nice feature. Then some of the other pairs, we have the hydroxyl. As you can imagine, adding the hydroxyl to an aromatic ring increases the solubility. For an aniline or NH2 type group, it's about neutral. It doesn't reflect it any. And then down here we have some halogen atoms added to a aromatic ring, and basically what we're seeing is that we're decreasing the solubility of these matched molecular pairs. So again, we can look at the chlorines and then click on the pair tab, and this will show the pairs within the chlorine by adding a chlorine. Okay, so these are sorted in order of descending change in the log of the solubility. And so what you can see is this top pair, by adding a chlorine in this position on the molecule, we actually increase the activity, or excuse me, increase the solubility of the compound. And maybe what's happening here is that the chlorine is pushing this amide group out of the plane, and that destabilizes the solid form and therefore increases the solubility of the pair. Okay, so now going on to back to the slides and discussing in detail changes in solubility. Okay, so Dr. David Miller, that's on this call, performed some more analysis, some more in-depth analysis than I did with the demo. So he used essentially the same set of molecules in the solubility data set. And so here we're showing the distribution log of the solubility, the molecular weight distribution for the 3,500 compounds, and then finally the predicted solubility, or excuse me, the predicted log P of the compounds using ADMET predictor. So from these about 3,500 compounds, 300,000 matched molecular pairs were generated using a similarity cutoff of 30 mismatched atoms. So this took about 15 minutes on a quad core, so a 4-core Intel Core i7 processor. And then the next step was to reduce these pairs using the cluster analysis. Okay, so the results are shown on this next slide. So we have some fairly simple ones. Of course, you'll recognize this addition to a chlorine to an aromatic ring on the second row. And this first row is essentially saying what happens when you add a methyl group to an alkyl chain. So here's the smirk string for that. So it's methylation of any aliphatic carbon atom. And if we look at these changes on the network viewer, we see that simply adding, as you add additional methyl groups to a linear chain, that pretty much just decreases the solubility. What confuses the issue a little bit is the branching. So while the methylation effect is clear on the solubility and that exhibits a clear trend, it is complicated due to branching. So that's just one simple example. As shown in my demonstration, you can extract additional pairs or specific pairs. In this case, we've looked at a single aromatic substitution of an aromatic ring. Okay, so you see the same kind of trends as I showed in the demo. Most specifically is that addition of heavier halogens causes a large and consistent change in solubility. So as you increase the molecular weight of the halogen, you decrease the solubility in a fairly straightforward manner. And I do want to mention this publication by Leach et al. From AstraZeneca that was published in 2006. And it's a very nice article, and if you read the article, you'll recognize some of the examples presented here in this webinar. If we compare adding a methyl group to an ethyl group, the ethyl group decreases the solubility more than the methyl group. Carboxylic acid, methoxy, and NO2, these were not shown in my demonstration because these involve more than one atom being added to the core. But you can see that when you add a carboxylic acid, it really, sometimes it increases solubility, sometimes it decreases. So the overall average is pretty much no change whatsoever. Adding a nitro will decrease the solubility in general. Okay. So going to the next slide, the general solubility equation relates the solubility to the hydrophobicity of the compound and the molecular weight, or excuse me, the melting point of the compound. And we can plot the average change in the log of solubility and the average change in the log P for these molecules as shown here. And what you see is just a trend as the additional atom becomes more hydrophobic, then the solubility decreases. So as you add the iodine, that decreases more, the solubility more than the bromine atom. Okay. So in another set of experiments to try and see what the effect of extending the context or the around the particular site is, David performed the following experiment. So first he identified all the minimal context transformations represented by at least 10 matched molecular pairs. And then for each transformation stored the average observed change in solubility. And then in order to predict the solubility of a single molecule, he took all the known solubilities of the neighbors for that molecule that are involved in the same pair. And then, so that was kind of the basis of the solubility, and then simply added the average solubility change for that matched molecular pair. So in this chart over on the left, this is the prediction as explained by these two steps here, and versus the observed value. We certainly get a nice trend, so the predictions we get the trend right in the observations, but the root mean square error is 0.73. And we can compare that to predictions from our global solubility model in ADMET predictor, and for these 2,500 compounds, it has an RMSE of 0.55. So certainly the global model outperformed the MMP model. So this was just a single atom context, so an aromatic carbon. Now, what happens if we kind of increase the context around that matched molecular pair? So here we want to recluster all the molecules in that same region, but be more specific. So here's an example down at the bottom, a biphenyl group, and you're adding the chlorine specifically in the ortho position in this biphenyl. So you have about 12 atoms you're matching the context with. And of course, you have a much lower number of pairs, in this case only 349, but using the same approaches on the previous slide, now the RMSE for these 349 compounds is 0.59. We can compare that to the predictions from ADMET predictor, where it's 0.60 from that global model. So we're definitely on par. So increasing the context increases the prediction, but the predictive accuracy, but the applicability domain is severely limited. Okay. So these next few slides just further illustrate that point. So here are the distributions where we have a very loose, or we're not including a large number of atoms in the context. If we then go and include a larger number of compounds, we have a much smaller variance in the distribution, and we have much more complex rules. This slide just illustrates going from 1 atom in the context to 4 atoms, 8, and 12 atoms overall. So here you see the distribution. We've also have the number of atoms and the standard deviation of this curve. And so going from 1 atom to 4 atoms, you haven't reduced it largely, and you've only reduced the standard deviation by a little bit. If you go to 8 atoms, you only have 95 pairs, and your sigma is 0.74. And then going to 12 atoms, your sigma is 0.63. So we've kind of shrunk up that distribution, and we could be more accurate when we apply this to an arbitrary molecule that would have the same context. Okay. So in this final section, I want to discuss using matched molecular pairs for predicting off-target interactions. So basically what we're using here is the maximum common substructure and the similarity approach to find compounds similar to our query molecule. If you have ligands that are known to interact with a certain receptor, and you find a match between your query molecule and one of these ligands, then your query molecule is likely to hit that same target. So here we just have 3 examples of different target properties or different targets to hit, and we're looking for matches in any one of these type targets. So let me just briefly show how we perform matched molecular pair search on a query molecule. This is the next version of MedChem Studio, and one of the features I want to point out is that you can now add a column that contains links to HTML pages. So we could click on this and it would bring up the link to that page, and David's kind of found matches from the World Drug Index and their corresponding wiki pages and added that to this. Okay, so a couple things we've done in this next version is we've allowed an option for using multiple processors on your machine, and that's simply under the Settings tab in the Advanced module. So here I've set it to 2. This is a dual-core processor as the maximum number of processors to use for these computations. Okay. We also have the ability to create a screening database, and basically what this does is it pre-computes some of the parameters necessary to perform fast searching. So this speeds up the searching greatly. So I've taken a subset of the World Drug Index of about 60,000 compounds and created a database of those in a previous step. Okay, so then I've highlighted the structure, and I simply go down to the Query tab, and I say Perform MMP calculation. It's going to search the World Drug Index 60K. It allows for up to 40 mismatched atoms and bonds, and I'm going to change the Tanimoto so that it only finds things that have a Tanimoto similarity of 0.7 above. And I'm going to ask it to import the search results into MedChem Studio. So I simply click OK. it goes through the searches, and as you see here, it's using 2 of the processors to compute the results. And it found 27 pairs. Now I simply click on the molecules pair tab to look at the pairs. So this molecule was already in the World Drug Index, or the database I searched. So the top match is this particular molecule. And we have a similarity score and number of mismatches column. In the second pair, we've replaced methoxy group with a methyl group, and we've removed a fluorine for this molecule. So overall, we have 27 hits with a similarity score of less than or greater than 0.7. Okay. Now let me go back to the slides. So in order to identify off-target potential interactions of compounds, we took the Chemical database and downloaded bioactivities and structures for 74 targets from the Chemical database. We identified 70,000 ligands from this set in step 1 that had an activity of under 30 micromolar. And then we took 750 approved drug molecules and used those as query molecules to search this 70K database. We had a similarity cutoff of 40 mismatched atoms and bonds. So this process took about 2 hours, and the choices of the ligands and the targets were from these 2 papers. So there's a recent paper in 2011 in Nature where they used a similar procedure, but use a different similarity technique. So this is just an example of some of the results. So we take diazepam as the query molecule. There were 774 hits in the 70K database, and this is simply a list of the targets. And so you go through these targets, and these are potential side effects that might be caused by diazepam. Okay, so if we look at 3 specific query and hits from those queries, and these were mentioned in this Nature Biotech article in 2007. So we see that methadone, which is a mu opioid agonist, matches this M3 muscarinic receptor. So luckily that this will also hit the muscarinic receptor. Imetine matches an alpha-2A adrenergic ligand, and then finally loperamide matches an NK2 neurokinin ligand with this. And so all of these were purchased and tested by this group, and was confirmed that they have activity at these particular targets. Okay. So in conclusion, the match molecular pair analysis can provide valuable insights into structure-activity relationships. MedChem Studio incorporates the scaffold-based molecular alignment to facilitate the analysis. The transformations can be extracted and then applied to other molecules in a straightforward manner. As far as using this QSPR, it's less valuable than traditional QSPR for quantitative property predictions, but as you increase the structural contact, the rules, in each rule you increase the performance and the predictability. So kind of in this last session, we looked at using MMP for predicting and rationalizing off-target effects. This was just a simple example, and additional work is needed to make the predictions a little bit more quantitative. So with that, I thank you and welcome any questions.
Thank you, Michael, for that very enlightening presentation. The floor is now open for questions. Once again, if you'd like to ask a question using your telephone, please use the hand-raising icon on your control panel and be sure to enter your unique audio PIN, or you may type the question in the questions pane found in the GoToMeeting panel on the right side of your screen. There are a couple of questions, and I have unmuted Bob Clark and David Miller. So Michael, David, or Bob, please feel free to respond with answers to some of these questions. The first is, when designing a drug molecule, we need to check the hemolytic activity. Which software tools could be used for this prediction in silico?
I'll let Bob answer that one.
If it depends a lot on how much data you have and what kind of data it is. If you have a limited amount of data, but it's mostly, for example, qualitative in the context of, in a similar molecular context that you're trying to check out, then you might want to use the MMPA analysis. If it's more diverse substructures, but not a lot of them, then I would recommend looking at the ADMET modeler software we have in ADMET Predictor.
Thank you, Bob. A second question, how does MMP analysis relate to MedChem rules of thumb? For example, adding a hydroxyl group increases solubility and reduces lipophilicity.
I think they line up very nicely with rules of thumb. So as you saw in the chart, adding an OH did increase the solubility and the lipophilicity of the pairs. So depending on what property you're looking at, these do correspond well with medicinal chemistry rules of thumb. But you can go more in depth with these, and that's the key. Typically, someone will maybe look at a series of data and only pick out the pairs that kind of validate their hypothesis in the first place. And with this tool, you can look at all the pairs so that you're not saying a certain change is due to a certain structural change in the ligand, just on kind of a biased approach of looking at a few of the pairs in the data set.
Thank you, Michael. Another question, in your experience, how big does a database have to be for MMP to be useful?
It depends what kind of property you're predicting. So in the AstraZeneca paper, they said that you need around 20 matched molecular pairs in order to get good statistics in order for that transformation to really be valid for an arbitrary molecule. The solubility data set was 3,000 compounds. So I think for property data, that's kind of the size that you need. If you're looking at structure-activity relationships and looking at maybe activity cliffs, the data set can be smaller, but it also depends upon how many pairs are in there. So you need 20 pairs from the AstraZeneca paper, but that's for property predictions. I think for ligand types, potency, you might be able to get away with fewer than that number of pairs.
Thank you, Michael. Another question, how literal does the shared context have to be? For example, can pyridyl isomers be included along with phenols to give the distribution of changes for argon hydrogen going to argon methyl or argon chlorine?
I think maybe that was supposed to be aryl hydrogen. Aryl chlorine.
Dave, could you answer that question maybe?
Yeah, there is a feature on, in fact, I don't know if I think you did show that dialogue, Michael, but on the options dialog where you are choosing the amount of context that you would like to require, in order to group two matched molecular pairs together into a cluster. There's a couple of options related to how specific the context has to be, and I'm trying to remember what the two options are. I think there's one related to ring topology, perhaps one related to aromaticity. I don't know that there's one that could actually allow you. Okay, so you have the screen up there. So I believe the first one is used to sort of allow all atoms and bonds to be treated as identical. So it wouldn't look at the atom type or the bond type. It would just sort of look for sort of the rough topology to be the same. And in fact, Michael, if you hover over that, there wouldn't by any chance be a wonder if because you're using a new version of the software, so it probably doesn't have tooltips yet. But yeah, I believe that those two options together would allow you to have a very sort of gross level of context that doesn't take atom type and bond type into account.
Yep, that sounds good. There's a lot of options of the program that I didn't show, and so that would be one that you'd want to try. And so from the solubilities, instead of just phenyl rings, you might look at pyridyl rings, et cetera, in the same pair. So that's something I can try later on.
Thank you, David. Thank you, Michael.
Mm-hmm.
Another question, when making the activity comparisons for a pair of molecules, are the bioactivity measuring assays restricted to being enzyme assays, or are they also available for whole cell assays? Or can whole cell assays be used?
I think you can use whole cell assays. I don't think that's a problem. It might be a little bit more difficult to interpret, but I think you could get some nice activity cliffs and rules out of using the cell-based assay.
Okay, now related to this, for the latter, for the whole cell assays, and this might be a little bit off topic, but how do you account for the difference due to transport-related properties versus site of action binding contributions to the overall observed bioactivity?
Yeah, that's the transport's going to obviously affect things. Maybe you can pick out molecules that don't get into the cells, I guess. Bob, do you maybe have some more insight on that?
I think it'll depend a lot on how broad, how wide a net you're trying to cast. If you have a lot of structural variation in your data set, then you're going to have potentially a lot of different effects that can be coming into play in the whole cell assays. In that case, you're just going to need more data. You need more examples so that you can map out each of those effects. But there's no intrinsic reason that either the matched molecular pairs or the QSAR, the artificial neural net models that you can produce with the ADMET predictor software, no intrinsic reason that they can't take care of multiple individual methods. It ends up just giving you a patchwork. As I said, the main problem is getting enough data to do a more complicated case.
Thank you, Bob. Thank you, Michael. This is somewhat off topic, but coming from a client, will there be in this next release of MedChem Studio a database query function? Could you also briefly touch upon what would be some of the additional new features we can expect? David?
Yeah, sorry, I was muted. So, Michael, did you show the database query feature?
Yes.
Okay, so that's.
So that was just
Yeah, so that's the idea of kind of pre-processing a set of structures like the World Drug Index, such that they can then quickly be used in a matched molecular pair type of screen. So you build the database once, and then you can screen against it, very quickly, many times in the feature. So that's going to be one of the primary new features in the software. There are several other features that are planned. The graphics being improved is one of the features. There's a lot of improvements to the sketcher. Looking at my here.
Minor 3D has also been updated so that there's more graph types and functionality available within the graphing package of Minor 3D. I think that the query tool has been enhanced also.
Yeah, we've vastly sped up the matched molecular pair analysis algorithm. So I think Michael showed some examples of generating pairs. That process was quite slow. It's a real full MCS-based approach to generating pairs, and so we've managed to speed that up by about tenfold, and that's in the absence of using multiple processors, which, like Michael said, is also going to be an option for that, and a couple other features that you'll be able to take advantage of multiple cores. Kind of related to that, we actually plan to release a 64-bit version of Metcam Studio as well, with a considerable amount of memory. You'll actually be able to utilize that memory and won't be limited to the 2 gigs of memory that you're currently limited to on your 32-bit system. So that's another update that we're planning.
Just one more comment. So a database query function, the other thing this could mean is maybe querying an Oracle or a SQL database, and I don't believe that's going to be included in the next version. Maybe David can comment on that.
No, that's true. So that is something that we had talked about doing, but I think that's going to have to wait until a subsequent.
Thank you, David. Thank you, Michael. One final question or comment is about this MMP feature and whether it's available in ADMET Predictor also, or just exclusively to Metcam Studio. So this is available just with Metcam Studio. We can follow up separately offline with the person asking this question about how to obtain a license to the Metcam Studio software. I don't see any final questions from the audience. So what I'll do now is ask Anu to make some final closing comments about where people can see us next. And once again, excuse me, thank you, Michael, Bob, and David, for your technical contributions today. Anu?
Yes, thank you. Thank you, John. I'm just trying to make my screen bigger. Okay, so yes, thank you, Mike, and thank you, John. These are the contacts of Mike, John, and myself. If you need to evaluate any of our software, if you have any questions related to any of the softwares, you could contact us, and I will be sharing this presentation slides with you, so you will have our contact details. And you can also see us if you are attending the 7th annual I2PC meeting in Pennsylvania. We will be exhibiting in the exhibit hall there. And also, if you're attending the Society for Toxicology annual meeting in San Antonio, Texas, Simulations Plus will be exhibiting at that event as well. And besides that, we are also holding 2 workshops this year. One is for the ADMET Design Cert and the Gastro Plus, which would be a big combination of all our product, pretty much ADMET Predictor, Metcam Studio, Gastro Plus, and some of the TDD Plus as well. So this is upcoming in March, and that's going to be in Boston, Cambridge, Hyatt Cambridge. So if you're interested in learning more of our products, please let us know. Either you can visit us on our website or just contact us individually, and we'll be happy to assist you further. Also, for the Gastro Plus, if you cannot make it in March in Boston, then you can attend the Gastro Plus workshop in San Diego, California. That will be in April. So we look forward to assisting you further. And with that, I hand over back to John for closing this presentation.
Thank you, Anu. That concludes today's webinar. If you missed any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. Thank you so much for joining us, and have a wonderful day. Take care.